All right, so next we're going to discuss bacteriophage. All right, in the last video I told you there were two types of capsids that were associated with viruses that infect mammalian cells. Those are the helical and the isohedral capsids. The capsid that is associated with bacteriophage is considered a complex capsid. All right, the complex capsid is seen just with the bacteriophage. All right, and in terms of those bacteriophage, they've got really kind of a unique shape to them. All right, so you're going to kind of have the head area here that's going to have your capsid, your genetic material is going to be inside, all right? And then in terms of our bacteriophage, they're a little bit more complex. You've got a sheath that's associated with it. You can kind of think of that sheath like a syringe, because essentially what these bacteriophage are going to do, they are going to inject that genetic material into the bacterial cell, all right? And so that sheath is kind of where that happens. You're going to have kind of at the bottom here a pin, and then the bacteriophage to me kind of look like they belong on the as part of the landing party on a moon mission, because they've got some legs really associated with them. These are the tail fibers, and that's essentially what will sit down on our bacteria to inject that genetic material into the bacteria. All right, so our bacteriophage, that's what I just drew here, and some people call it bacteriophage. Either's fine. I've always learned it as bacteriophage. All right, these are going to specifically infect bacteria. All right, these will not infect eukaryotic cells. All right, and in terms of that, that's one potential advantage and one potential route that is being investigated as far as an alternative to antibiotics. Because antibiotics, with the antibiotics we have, the bacteria are adapting to them, and they're becoming more and more resistant to the antibiotics we have in our arsenal, which means we either need to make more antibiotics or we need to find a different way to kill bacteria. The bacteriophage are potentially one way to do that, all right? What's problematic with bacteriophage, using them to treat infections, is right now they will infect not only the pathogenic bacteria, but they'll also infect those good bacteria that are helping keep your gut going. So in terms of that, that's one of the downsides that they're working on, all right? But the bacteriophage is going to be essentially a virus that will infect bacteria. So I'm going to draw a little bacteria here in pink, all right? And since I did our bacteriophage in green here, we'll make it green here. And essentially what that bacteriophage does first, it has to attach to the bacteria itself, all right? And we'll draw, this is going to be the DNA for our bacteria, and we've got DNA within our bacteriophage, all right? So what's going to happen, that bacteriophage is going to find the bacteria, it's going to set down on it, it's going to attach. The second step that bacteriophage now is going to inject the DNA into the bacterial cell. That's where that pin comes into place. It kind of penetrates the cell wall, the cell membrane, allows that DNA or genetic material to be injected into the bacteria. All right. Then what's going to happen? All right. We've got our DNA here. We're going to have replication of the genetic material, because what ends up happening with viruses, and one of the things that viruses do is they use the host cell machinery for important processes. Those important processes are going to be DNA replication, transcription, and translation. So what's going to happen in that third step with replication that viral genetic material now is using the ribosomes within that prokaryotic cell to make new protein coats. It's using the polymerases for DNA replication to make more copies of the viral genome. All right. Then what we're going to have happen in our fourth step is those viral particles now are going to be assembled. And what's going to happen, those viral particles are going to kind of squeeze out all of the other stuff that's going on inside of that bacterial cell. And that's going to leave us with our last part, which is going to be lysis. So what's going to happen? That cellular machinery within the bacterial cell is going to make so many viral particles. What is going to happen is our bacterial cell is going to break open, or it's going to lyse. So it's going to burst open and it's going to release the viral particles into the environment. If there's bacteria nearby, those bacteriophage can attach to that healthy bacteria. That same process can happen again. Okay? So in terms of our bacteriophage, the cycle that I have here is what's known as the lytic cycle. All right? Lytic getting to lysis and breaking open of that bacterial cell. All right? There's a second way that the viral DNA can exist within a bacterial cell. And I've kind of used up a lot of my board here. But I'm going to erase this part, and I'm going to talk about the other way that those viral DNA can exist within a bacterial cell. And that's known as lysogeny, or the lysogenic cycle. All right? The lysogenic cycle doesn't kill the bacteria immediately. All right? What happens with the lysogenic cycle, that bacteriophage is still going to attach, it's still going to inject its DNA. But what ends up happening with the lysogenic cycle is if we have our bacterial DNA here, and our viral DNA here, that viral DNA ends up integrating into the bacterial DNA. So you aren't going to lose any of the bacterial DNA, but, now that's in green, that viral DNA kind of incorporates itself into the bacterial's genome. And it kind of sits there silently, all right, until the environment changes or something happens. Then what will happen, that lysogenic cycle will be triggered to enter the lytic cycle, and you'll get production of bacteriophage again, all right? It's not well understood why the viral DNA would go towards the lysogenic versus lytic, but it is being studied. The lysogenic just kind of allows essentially that viral DNA to exist within the bacterial cell. The advantage here, evolutionarily, for that virus is that when that bacteria now undergoes binary fission, instead of having one bacterial cell that has viral DNA in it, you'll have two, okay? So all of the offspring of the bacteria that divide when that viral DNA integrates into the genome like that will have that viral DNA associated with it. So you'll have more bacterial cells that will actually go into the lytic cycle, all right? So in terms of that, those are our bacteriophage. I want you to understand the difference between what happens between the lytic and the lysogenic cycle, the steps that happen that are associated with the lytic cycle. If you have any questions, please let me know.